Hello and welcome back. This is our fourth and final video in our video series for chapter 6, which is expression of biological information. I'm Miss Delia, and in this video, we're going to go into the final subtopic, which is 6.4 gene regulation and expression using the example of a lac operon. If you're ready, let's get started. So here are the learning outcomes for this subtopic, so that by the end of this lesson, I hope you would be able to explain the concept of operon and gene regulation, state the components of operons, explain the components of the lac operon and its function in E. coli, and last but not least, explain the mechanism of the operon in the absence and presence of lactose. So this is basically what an operon is. To look at it more closely, an operon is a functioning unit of DNA containing a cluster of genes under the control of a single regulatory signal or promoter. Dalam arti kata lain, apa itu operon? Operon adalah sekumpulan gene atau satu cluster gene di bawah kawalan satu promoter ataupun regulatory signal. So asalkan operon ini di-on kan, as long as it's turned on or switched on, all the genes that are in this cluster will also be transcribed and then translated. Ataupun kita boleh katakan all the genes will be expressed if this operon is turned on. If it's turned off or switched off, then everything in this operon will also switch off. Mari na kita tengok. Okay, these genes will be transcribed together into an mRNA strand and are either translated together or separately in the cytoplasm. Jadi bukan saja mereka duduk bersama dan di-on bersama, tapi bila dia kena translate jadi mRNA, dia akan bergabung dan duduk pada mRNA yang sama. The example that we're going to use in this chapter is lac operon. Contoh yang kita gunakan adalah lac operon. Lac operon ini dijumpai dalam bacteria E. coli. So it's found in E. coli and it will be induced by the existence of lactose in human colon. So in case you're not aware, our stomach, our digestive systems have bacteria inside. We have bacteria in our guts, and one of them is E. coli. Escherichia coli ini adalah jenis bakteria yang boleh dijumpai dalam usus kita. Okay, so bakteria ini ada lac operon. Bila kita minum susu mengandungi lactose, itu akan mempengaruhi sama ada operon ini di on ataupun di off. Okay, so let's have a better look at the lac operon. So this is something that is introduced by Jacob and Monod in 1961. The background is that E. coli lives in your human colon and it helps to break down lactose that it finds in our digestive system. So when we say break down lactose, it is carrying out lactose metabolism. Jadi E. coli itu akan gunakan enzim-enzim dia untuk ambil lactose dan memecahkan dia kepada glucose dan galactose. It's metabolizing lactose into glucose and galactose. The proteins involved in the lactose metabolism in E. coli are beta-galactosidase, beta-galactoside permease, and beta-galactoside transacetylase. So ini adalah 3 enzim yang digunakan oleh E. coli untuk metabolize ataupun untuk hadam lactose dalam badan. Okay, so let's look at the first enzyme. The first enzyme, beta-galactosidase. It will hydrolyze lactose to glucose and galactose. So inilah enzim yang memecahkan lactose kepada 2 monosaccharide ini, yaitu glucose dan galactose. The second enzyme, galactoside permease, is a membrane-bound transport protein that increases the permeability of the cell membrane and pumps lactose into the cell. Apa maksudnya? Ini adalah transport protein yang dijumpai pada permukaan sel membrane. Function dia adalah untuk membawa lactose ke dalam sel. Dia memudahkan lactose untuk masuk ke dalam sel E. coli. And thirdly is the enzyme beta-galactoside transacetylase. It is an enzyme that transfers an acetyl group from acetyl-CoA to beta-galactosides. But its novel function in lac operon remains unclear. Kalau di luar lac operon, kita tahu enzim ini fungsi dia adalah untuk memindahkan acetyl group daripada acetyl-CoA kepada beta-galactosides. Tapi fungsi dia yang spesifik dalam lac operon ini kita belum dapat tahu. Tapi kita tahu dia digabungkan bersama dengan dua gene yang lain. Dia ditranscribe sama-sama dengan mRNA untuk beta-galactoside and galactoside permease. So, mungkin ada kaitan dia. Cuma kita tidak tahu sejak kaitan dia apa for now. Okay, and then we'll talk a bit more about the lac operon. The lac operon referring to everything in this diagram, yeah? The function of this thing in E. coli is that it functions for the transport and metabolism of lactose. Sebab gene-gene ini mengandungi gene untuk hasilkan enzim untuk metabolize lactose. Jadi fungsi utama dia adalah untuk transport and metabolism of lactose by producing enzymes that break down lactose for energy or source for other use. This operon will be activated or switched on when lactose is present. Okay, bila lactose ada, dia akan on. So let's look at the components of the lac operon and its functions. Apa yang ada dalam lac operon kita? Lac operon E. coli. The first component is the regulatory gene ataupun LAC-I yang di sini pertama. LAC-I will contain the gene to synthesize repressor protein. Ini akan hasilkan mRNA LAC-I. LAC-I mRNA will produce repressor protein. The second component of the lac operon is the promoter. Over here, it is the green plus yellow area. This is the attachment site for RNA polymerase. RNA polymerase yang tukang buat mRNA itu akan mula melekat di sini, di promoter. Operator. Operator is the attachment site for repressor. Repressor akan melekat di operator. And the structural genes. So the structural genes here are LAC-Z, which is the gene to synthesize beta-galactosidase, LAC-Y, the gene to synthesize beta-galactoside permease, and LAC-A, the gene to synthesize galactoside transacetylase. Okay, so you have, once again, you have your regulatory gene, you have your promoter, your operator, and your structural genes. Okay, so far so good. Those are the components. Now, let us see how they function. Mari kita tengok macam mana dia berfungsi. So this is how the operon looks like when there is no lactose in the system. Okay, bila tiada lactose dalam sistem, LAC-I will be transcribed and translated into repressor protein. So this one, nothing to control it to turn on or turn off. So it's always on. Okay, the regulatory gene is produce mRNA. mRNA hasilkan repressor. Repressor protein will then bind to the operator. Repressor protein ni bila dia boleh bekerja, dia akan pergi dan menekat kepada operator. Bila dia menekat kepada operator, RNA polymerase cannot bind to the promoter. It prevents RNA polymerase from binding to promoter. RNA polymerase ni mau melekat di DNA, tapi sebab ada repressor protein halang dia tidak dapat lekat dan tidak buat transcription. So there is no transcription of structural genes and no enzyme is produced. Therefore, without lactose, operon is not active. We can also say operon is now switched off. What happens when we do have lactose? Okay, mechanism of the lac operon with lactose, it will look like this. If the lactose is present, some of the lactose will turn into its isomer, which is L-lactose. L-lactose adalah isomer, bermaksud dia lactose juga, cuma susunan molekul dia berbeza sedikit. Okay, sesetengah lactose akan diubah menjadi L-lactose. Pada masa yang sama, LAC-I masih kena transcribe. LAC-I is transcribed and translated into repressor protein. Okay, dia kena transcribe mRNA, hasilkan repressor protein. Because now our environment has lactose and allolactose, the allolactose will bind to the repressor protein. The repressor protein will change its conformational shape. Okay, dia akan melekat di sini dan bentuk repressor protein itu terus berubah. Disebabkan bentuk dia sudah berbeza, kalau kamu tengok di gambar raja ni macam dia melengkung sedikit kan. Okay, as long as the shape is changed, the conformation is changed, it cannot bind to the operator. Repressor protein cannot bind to operator. Now, the RNA polymerase can bind to the promoter. Dia boleh melekat sudah. Jadi, asal RNA polymerase ni boleh lekat, dia lekat kepada promoter dan dia akan start transcription. The RNA polymerase will now do its job of transcribing the genes. So you will have transcription of structural genes occur. Your enzymes will be produced and the operon is active. Ataupun we can say the operon is switched on. Okay, so just a quick summary. This is what the operon looks like when it's switched off and when it's switched on. Why do cells need operons? Because sometimes you don't need the enzyme being produced all the time. Kalau enzim itu sentiasa dihasilkan, transcription dengan translation balik-balik dihasilkan, berjalan, itu akan memakan tenaga dalam sel, membazir tenaga sel kita, dan membazir bahan-bahan dalam sel kita. So to reduce waste of energy and materials in the cell, some operons are used to turn off certain genes and only turn them on when necessary. Okay, so this lack operon is kind of the main gist of this subtopic. Memang tumpuan utama subtopic ini adalah pada lack operon. Tapi jangan lupa, kamu juga perlu faham apa itu definisi operon. Don't forget the definition of an operon. Don't forget what are the components of an operon. The regulatory gene, the promoters, the operators, and the structural genes. And don't forget the components of this lac operon that can be found in E. coli. If you can give a simple explanation of how they can turn on and turn off, then you're already doing well for this chapter. Okay, so we also have this video on gene regulation and expression for lac operon.
The lac operon has several parts to it, but we're going to go through each of these parts and talk about their functions.
YouTube,
in prokaryotic cells, genes are often found in groups called operons. So actually what we tend to see is not just one gene on its own followed by another gene. We can actually find one or often more than one gene grouped together into a particular region.
Okay, so that is the end of 6.4. What should you now? What should you do now? You should go and make your own notes for 6.4 and also answer the Google form quiz for this subtopic, which you should be able to find in your Google Classroom. I think it's combined with 6.3 in the same Google form, so it should be you should be able to complete it together. Okay, so that is all for me. I will see you again in the next lesson. Thank you very much for your time and attention, and happy learning. That is the end for chapter 6, and I will see you again soon. Bye!